In DNA, Kenyon encountered a molecule with a property he could not explain through natural processes. For locked securely within its double helix structure is a wealth of information in the form of precisely sequenced chemicals that scientists represent with the letters A, C, T, and G. In a written language, information is communicated by a precise arrangement of letters. In the same way, the instructions necessary to assemble amino acids into proteins are conveyed by the sequences of chemicals arranged along the spine of the DNA. This chemical code has been called the language of life, and it is the most densely packed and elaborately detailed assembly of information in the known universe. By definition, natural selection could not have functioned before the existence of the first living cell, for it can only act upon organisms capable of replicating themselves, cells equipped with DNA that pass on their genetic changes to future generations.
Without DNA, there is no self-replication. But without self-replication, there is no natural selection. So you can't use natural selection to explain the origin of DNA without assuming the existence of the very thing you're trying to explain.
With computer animation, we can enter the cell to view this remarkable system at work. After entering the heart of the cell, we see the tightly wound strands of DNA, storehouses for the instructions necessary to build every protein in an organism. In a process known as transcription, a molecular machine first unwinds a section of the DNA helix to expose the genetic instructions needed to assemble a specific protein molecule. Another machine then copies these instructions to form a molecule known as messenger RNA. When transcription is complete, the slender RNA strand carries the genetic information through the nuclear pore complex, the gatekeeper for traffic in and out of the cell nucleus. The messenger RNA strand is directed to a two-part molecular factory called a ribosome. After attaching itself securely, the process of translation begins. Inside the ribosome, a molecular assembly line builds a specifically sequenced chain of amino acids. These amino acids are transported from other parts of the cell and then linked into chains often hundreds of units long. Their sequential arrangement determines the type of protein manufactured. When the chain is finished, it is moved from the ribosome to a barrel-shaped machine that helps fold it into the precise shape critical to its function. After the chain is folded into a protein, it is then released and shepherded by another molecular machine to the exact location where it is needed.
This is absolutely mind-boggling to perceive at this scale of size such a finely tuned apparatus, a device that bears the marks of intelligent design and manufacture. And we have the details of an immensely complex molecular realm of genetic information processing. And it's exactly this new realm of molecular genetics where we see the most compelling evidence of design on the Earth.